It stands for sarcoma. SARC is elevated in many different tumors. SARC is activated by growth factors. SARC then turns off cell death, which leads to cancer cell survival. It helps develop blood vessels to the growing tumor, and it also helps with cancer cells invading other parts of the body. We suggest a new pathway to cancer then, as PA is elevated in cancers. Many other researchers have shown PA is also elevated, just like SARC. We found that PA can bind and activate SARC and suggested this as being a new pathway to cancer. So continuing that kind of work, we have this model. Here is SARC, it's off. All these different domains are bound up. There's weak bonds all over this thing, keeping it bound up and turned off. It does have a lipid tail. But then this SARC will isolate itself from the rest of the membrane in what are called membrane rafts. So we suggest that certain, by our binding studies, PIP2 and PI4P are here in rafts only, and that they will localize this SARC to a specific section of the membrane called a membrane rafts. Membrane rafts are required by hormones and in fertilization. So in fertilization then, we have the production of this weird phosphatidic acid. It's very highly charged, it's very unusual in its structure, and a guy named Miguel Pons in Barcelona has found other weaker anionic lipids will bind to these domains of SARC, SH4, unique lipid binding region, and the SH3 region. These, especially SH3, is found on many different proteins. But we suggest then that PA is increasing at fertilization. We've measured its mass, and that based on his work and some of our work, we suggest that it binds and causes this unfolding of SARC, where now this thing is turned on. SARC can do stuff. It can basically cause the fertilization events, phospholipase C activation, but this also is a cancer-causing enzyme. This could be another way, if you increase PA, of causing cancer. So this conformational change is what my current master's student is studying over at the medical center, doing NMR studies, looking at the structure using this part of the enzyme and how it will be affected. The structure will be changed, the conformation changed by binding to PA. And that's Chase Riedel. So something that Colby did was very basic. It's just kind of cool. Normal fertilization on the left. Here's FIPI. We inhibit the production of PA with this FIPI. So normal fertilization. We've got, should be turning on. And over on the right, not much is happening. Over here on the left, this is normal fertilization. And a sperm binds there, a little bit of calcium is released, then the sperm is unsuccessful, but it's staying there. This is called like a kiss and release kind of a thing with neurotransmitters. But the sperm stays there, it finally succeeds. This glowing area is high calcium, calcium is being released, and then there's a calcium wave that travels across this whole large egg. The sperm is so small you can't see it, but it has to activate all sides of this monstrously large cell. So there's a calcium wave that's generated. It's true in the frogs, it's true in humans. But when this restarts here, we'll see that Colby not only was able to describe that FIPI, yes, it does inhibit the calcium release, PA production, you inhibit it, you inhibit calcium release, but in normal fertilization, you also can find things that other people didn't see. And if this replays, which it should, right over here, if you look closely enough, and you can't really see it, we've got better videos, there's actually a rotating disc that will start and appear here and then rotate in an outward way to produce the egg. It's hard to see, but we've got better videos where that rotating disc is there. So that was a new finding, and it's totally against the model for this calcium wave. So, as Yogi said, you can observe a lot by just watching. And Colby and I published a paper last year where it's a new mechanism for that calcium wave, that is a rotating disk of calcium, something we were kind of interested in. New mechanism. Jason, who's here also again, looked at tyrosine kinase inhibitors that should inhibit all of the production of calcium release by PA. Turns out it's only a partial inhibition. Won't go through that. But now we've got this idea that this lipid PA will act directly on SARC, as we've said. That could also affect cancer, and it also stimulates this pathway to calcium. But we found that PA also binds, or not binds, we haven't shown that, but activates PLC activation. And so there's two effects of this PA. And it turns out when we suggested that, look back in the literature, there were tons of papers from the 1980s showing that PA binds and activates PLC directly. So again, it's that belt and suspenders idea that Mother Nature has backup systems. This is the fast way, this is the way it normally occurs, but if you inhibit that, then this pathway takes over and still provides you with enough calcium to cause fertilization. That's our current manuscript. So I'm going to start with this one right here, and that's just, again, our current model. We've expanded those steps. All the red steps are things that we've talked about in this talk that we've contributed. And I'm going to leave it right here. Thanks for your attention.
So we have time for one question, and there's, so make it a good question.
Confused everybody. It's always a good teacher. Dr. Petco.
What's next for you?
Travel. As you know, I'm going on phased retirement, and so I'll still be one committee, one class a year, and I'll hopefully have more time to finish the 2 papers that I've got writing, and also finish our current experiments. We've shown that PA causes SARC to cycle. SARC goes up and down in terms of its activity. Fertilization involves the cycling, so we're thinking that SARC may cycle because it activates its own inhibitor, a tyrosine phosphatase, and then that would reduce the activity, but your signal is still there, fertilization or PA, so it increases again. So why SARC cycles? Maybe that's where, again, related to cancer, related to fertilization, but that mechanism is what we've been working on. Yes, Jason?
Do you think some of these lipids or pathways play a role in like exosome signaling?
Yeah, so that's the biggest field now. Fertilization is known to have exosomes. So exosomes are, if you have a cancer cell or a normal cell, they are spewing out little tiny vesicles. They're spewing them out, they get into the bloodstream. So now you can take a blood sample and say, oh, you have pancreatic cancer. Why? Because your exosomes have proteins and lipids from the pancreatic tumor. So you don't have to do scanning, you just do a blood sample and look at the exosomes. So that's how it's also thought that one cell will send off signaling molecules, signaling lipids in this little tiny vesicle that then gets into the bloodstream and spreads around. That's kind of a scary thought that you have a cancer cell putting off little vesicles that might cause cancer in other parts of the body. Not an entire cell with the nucleus and all the other parts of it, but a tiny little vesicle. So exosomes are known to be an effect in the fertilization. People suggest that sperm are releasing exosomes that will affect the egg. The egg is known after fertilization to release exosomes. What they are doing is releasing exosomes that have the sperm receptor. So when one sperm binds and gets into the egg, you don't want any other sperm coming in with excess DNA. That'll cause cell death. So the first sperm will activate the egg. The egg then releases exosomes that carry away the sperm receptor. So yeah, exosomes, big field in lipid biology now. Mike?
Of your many amazing contributions in teaching, research, and service, what are you most proud of, looking backwards?
Well, when you see people showing up that worked in my lab 20, 30 years ago, I think it's the impact that you have on people that really means the most. And I'm also proud, of course, that we've introduced new ideas about fertilization or glucophage, but the point is that it's what do you think about when you're at home after a day at work? You think about the people, in addition, of course, to the results. But yeah, I'd say that's a big part. It's been fun to work with people and come up with these fun ideas. So for me, it's been, again, a great career. It's really what I wanted to do, and I was lucky to be able to achieve it.
Well, that's probably a great one to move on. So let's thank Brad again for such a great. Thank you all for coming again. No need to stop the questions now. Keep the conversation going. There's refreshments out there. And thanks.